Good afternoon, everyone, and thank you for joining us today for a Science of Childhood Cancer lecture from St. Jude Children's Research Hospital in Memphis, Tennessee. It's my pleasure to introduce today's speaker, Dr. Scott Armstrong from the Dana-Farber Cancer Institute in Boston, Massachusetts. So just a brief introduction to Scott's background and education. So Scott received his bachelor's degree in chemistry from the University of Oklahoma. Then he went off to earn an MD degree and a PhD degree at the University of Texas Southwestern Medical School, where he was a graduate student with Brown and Goldstein at Southwestern. Following that training, he then did his internship and residency as well as a postdoctoral fellowship at the Dana-Farber Cancer Institute and Boston Children's Hospital. For his research studies, he was a fellow with Stan Korsmeyer. Following that, he took an independent position at Dana-Farber and Boston Children's Hospital, as well as Harvard Medical School, where he rose through the ranks. But in 2012, he then moved to New York City to take a position at Memorial Sloan Kettering Cancer Center. However, he was in New York City for just 4 years and was recruited back to Boston with a position at Harvard Medical School, as well as having an appointment at the Broad Institute, supported by Harvard and MIT. So in Boston, he is chairman of the Department of Pediatric Oncology at the Dana-Farber Cancer Institute, as well as associate chief of the Division of Hematology and Oncology at Boston Children's Hospital. By way of honors, Scott is a fellow of the American Association for the Advancement of Science, as well as an elected member of the National Academy of Medicine. Scott's published over 200 papers, with many of these in the very top journals, and his research focuses on understanding the molecular mechanisms that underlie altered gene expression in cancer, as well as the development of novel therapeutics against cancer based on this knowledge. So it's my pleasure to have Scott with us today and to present a lecture on his current research. So without further ado, I turn the floor over to Scott. So Scott, welcome, and thank you very much for joining us today.
Thank you, Richard, for the nice introduction. That introduction gets longer as we get older. It is a reminder. All right. Thanks to everyone for joining. It's my pleasure to speak for the next 50 minutes or so about some of the latest work that we've been doing focused on the role of various chromatin complexes, mostly in leukemia, but I'll talk a little bit about some other cancers at the end. I'll also focus significantly on the drug discovery and drug development part of our work, mostly because I think this is likely a very broad group of people with broad interests, but certainly I'm happy to get into the details of what mechanisms if people would like to do that, and certainly I'll do that in the context of some of the molecules. So these are disclosures. I do work with a number of biotechs and pharmaceutical companies to help try to push these molecules forward. The ones that are relevant for today's discussion are C4 Therapeutics, that's developing targeted protein small molecule degraders. I'll talk about one of those at the end. And Syndex Pharmaceuticals, that's working on development of a menin inhibitor that I'm also going to talk about. So to put chromatin and epigenetics in the broadest context, as most everyone knows, we start in mammalian development with a single cell, a zygote, that gives rise to all of the cells required for the fully developed mammal, all of the tissues, all of the different organs That process can take from many days to months, depending on the mammal. And it does this. This development happens with the DNA sequence largely remaining the same. I say largely because, as you know, the immune system does rearrange a bit, and hopefully that's happening in many of us as we've been vaccinated for the pandemics that's keeping us at home. But this process of turning genes on and off during development is tightly regulated, highly complex, and clearly important for the development of pediatric cancers. The major proteins and complexes that are controlling this process are transcription factors and the chromatin-associated or so-called epigenetic complexes that directly modify and or rearrange or remodel the chromatin. Just a picture here of RNA pol 2 finding its way to open chromatin. Some of the chromatin complexes that we've been interested in, many people are interested in, are shown here, the types of complexes. They come in many different types, but broadly, the so-called Reiter complexes, these are the enzymes that modify place acetyl or methyl groups on histones. The erasers that remove those modifications, so these modifications are highly dynamic. And then those modifications provide something of a landing pad for the readers and proteins and the associated complexes. What's not shown here and probably binds via these readers are chromatin remodeling complexes that I'll talk briefly about as well. So probably the best defined system for studying development is the blood system. And that's because for decades, investigators have been defining the various cell types in the developing hematopoietic system, mostly in the mouse, but also in humans and other organisms. And a schematic is shown here, and I'll just go briefly over this. It's relevant for some of the discussion later. As most of you probably know, all of our blood cells arise from this single cell type, long-term hematopoietic stem cell, that's able to self-renew or make copies of itself while also giving rise to more differentiated downstream progenitors like are shown here in the middle, or so-called multipotent progenitors. And each step along the process, there is a change in gene expression program that's expressed, controlled both by transcription factors and chromatin complexes. And each time the cell makes a decision, it commits more and more to a single fate, and ultimately you get all of the different cell types shown here. I'll talk about these granulocyte macrophage progenitors a bit, so just to point that out. And as I mentioned, chromatin complexes are playing a role in this, and the complex we're particularly interested in is shown here on the right, the MLL complex. I'll talk more about that in a minute. And the reason that we became interested, or I became interested in the MLL complexes, is the result of the translocations in the MLL1 or KMT2A gene that are frequently found in pediatric leukemias, also found in adult leukemias, but more commonly or frequently in pediatrics. And this is just to show the first description in 1979 by the cytogenetic analysis here of a 411 translocation. At the time, it wasn't really known exactly what that meant, but it was recently shown, soon thereafter shown to be a recurrent abnormality. And then throughout the mid-'80s, a number of prominent investigators, Janet Rowley, Claire Boonfeld, Dr. Pui at St. Jude, a number showed that these were both recurrent and associated with particular clinical characteristics. That is, infants with leukemia very frequently had these MLL rearrangements, and that predicted a poor prognosis. It was also shown in the 80s that MLL translocations can be found in AML, and now we know somewhere on the order of 30% of pediatric AMLs have MLL rearrangements. It's less common in adults, but certainly still happens. And then in the early 90s, a number of groups with the progression in molecular techniques were able to clone the breakpoint. Again, Janet Rowley, Stan Korsmeyer, Carlo Croce, and Eli Kanani, and Mike Cleary's group, all about the same time, defined the MLL1 gene as the target of the various different chromosomal translocations or few that can be identified in leukemias. And we now know that the wild type MLL1 protein, initially shown by Stan Korsmeyer's group in a knockout study in mice, controls homeotic gene expression or Hox gene expression during both axial skeleton development and hematopoietic development. And that really solidified it as the mammalian homologue of a previously defined gene in Drosophila called trithorax, and really showed us that this is a central regulator of development in mammals, much like it is in flies. And then Mike Cleary and Terry Rabbitt's group showed really for the first time that the actual MLL fusion protein that's generated as a result of this translocation is indeed oncogenic via both knock-in mouse models from the Rabbitt's lab and a retroviral model initially developed by Mike Cleary's group, a model that many of us have taken advantage of over the past 20 years. So the wild-type MLL protein, the schematic is shown here. It's a number of domains. I won't get into the details of all of them, but just a relevant domain here at the end, or at least the domain that really showed us that this was a chromatin regulator, is the C-terminal set domain that has the histone H3 lysine 4 methyltransferase activity. So that part of the MLL modifies histone H3 on lysine 4 with a number of methyl groups, probably trimethylation. And that is associated with active gene expression. However, when the MLL fusion is, or the translocation occurs and the fusion is generated, the N-terminus of MLL, shown here, is fused to the C-terminus of what's now some over 60 or 80 different fusion partners. But the N-terminus of MLL is the consistent part. And this just shows the breakdown of some of those translocation and fusion partners. So in the early 2000s, this is whenever I was in Stan Korsmeyer's lab, and with the development of microarrays, I was interested in what it was, or which gene expression programs were active in MLL rearranged leukemias, and one of the schematics is shown here. We published that in 2002, right about the same time that Jim Downing and Bill Evans published their landmark gene expression study as well. And a number of interesting things came out of this, but one that's particularly relevant for today is that the Hox genes shown here, each one of these bars is an individual leukemia sample, are highly expressed in the MLL rearranged leukemias compared to, in this schematic, other acute lymphoblastic leukemias, pointing us to the fusion protein as likely being a gain of function since we knew the wild type MLL controlled Hox genes. That at the time was not perfectly clear, and certainly not clear in human samples, and this really set the stage for many of the studies that followed afterwards. So I've already given you the background, a little bit about the background of MLL, so I'll skip this, but just to remind you that this N-terminus of MLL is maintained in the fusion, the C-terminus is lost. So it's not immediately obvious why that would be a gain of function, since the part that positively regulates gene expression is here in the C-terminus. So then we continue to be, and we're at that point interested in what is it that the MLL fusion protein does at the developmental level to cells, in this case in the hematopoietic system. Another way to say, what is the cell of origin, or what can be the cell of origin of MLL fusion driven leukemias, and what happens when you go from a normal hematopoietic either stem or progenitor cell to leukemia. And this is going to summarize a bunch of studies. What we were ultimately able to show is that introduction of the MLL fusion either into long-term or hematopoietic stem cells, or more differentiated or differentiating myeloid progenitor cells like this granulocyte macrophage progenitor, would lead to the development of a full-blown aggressive leukemia. And we could isolate the so-called leukemia stem cells out of that leukemia and compare them back to the normal populations, and found that this leukemia stem cell is essentially a progenitor, looked very much like this granulocyte macrophage progenitor, that's inappropriately expressing gene expression programs like the Hox A cluster genes, MEIS1, and a number of other stem cell programs that are usually on in the stem cell and are getting reactivated in the context of this progenitor by the MLL fusion. And given all the gene expression that's done in mouse models and human leukemias, we now have a really good understanding of the details of the gene expression abnormalities seen in these leukemias. So the question, obviously, from a therapeutic standpoint is, how does the MLL fusion disrupt normal transcriptional control to drive or maintain leukemia development. So that takes us to the chromatin-associated complexes that are likely playing a role here. And I've already told you a little bit about MLL. This is the wild-type complex. The normal MLL1 is a histone H3 lysine 4 methyltransferase. And again, these are the normal complexes during normal developmental control of gene expression. And this complex has at least 7 or 8 other proteins associated with it. I'll talk about the details of those in a minute. The super elongation complex, which is a large complex that with an essential enzymatic component is a kinase that phosphorylates RNA polymerase on its C-terminus, CDK9, and the DOT1 complex, another histone methyltransferase that modifies histone H3 on lysine 79. This modification also associated with active gene expression. The reason these 3 complexes are relevant is, well, 1, MLL is here, and 2, many of these proteins, if not most, the ELL, AF9, AF4, AF10, have all been shown to be translocation partners or fusion partners with the MLL in leukemia, leading to the hypothesis that what's happening with the MLL fusion is essentially you fuse together inappropriately the MLL complex and the superelongation complex and or the MLL complex and the DOT1 complex. And many studies have shown indeed these proteins are in complexes together, and there's still active study going on to understand exactly which proteins do and what. But that then obviously leads to the question as to whether or not any of this can be targeted to reverse leukemogenic gene expression as a potential therapeutic opportunity. So things or proteins in these complexes that immediately stand out or where there has been some drug development are DOT1. I'm going to talk briefly about that. CDK9 is a kinase, so there are certainly CDK9 inhibitors that have been developed. And then I'll talk a bit in a bit of detail about small molecules that disrupt the interaction of MLL and menin. So MLL1 and menin form a direct protein-protein interaction. You probably heard about BRD4 inhibitors. I won't talk about those today. There are also small molecules being developed to target WDR5, which is part of the MLL complex over here. So a lot of interest in this space in terms of small molecule development. So in late 2000s, around 2010, Catherine Berndt, then a hem-onc fellow in the lab, developed a DOT1 knockout mouse where she was able to inactivate DOT1 genetically and demonstrate that the gene expression that was most sensitive to that, so you transform mouse cells with MLAF9, inactivate DOT1, the genes that were most sensitive to that were the genes, many of the genes where the MLL fusion is directly bound to chromatin. The first evidence that perturbing these chromatin-associated complexes really could lead to selective changes in gene expression. And that at the time, it still continues to be a question, but at the time we didn't know what would happen if you inactivated a chromatin complex that's bound to tens of thousands of places across the genome. But it turns out that when you inactivate DOT1, and I'll show you similar things with small molecules to other targets, that you get relatively selective changes in gene expression. The molecular mechanisms of that are still not, how you get selective changes still not particularly clear, and certainly an interesting question in the field. So with that in hand, at the same time we were developing small molecules that target DOT1. They're shown here. This is a small molecule from Epizyme called EPZ5676 bound to the active site of DOT1. And we were able to show that those small molecules significantly, but not identically, phenocopied the genetic inactivation. And that what happens when you inhibit DOT1 is if MLAF9 here is bound to DOT1 through AF9, you inhibit the enzymatic activity, you see repressive mechanisms like deacetylases and ultimately methyltransferases that place repressive methyl marks starting to show up at the genes where the MLL fusion was bound. And we were able to show that indeed what DOT1 is doing is preventing repressive mechanisms from shutting off gene expression that should be turned off at that particular stage in hematopoietic development. So this moved pretty rapidly into phase 1 clinical trials, this particular molecule. These phase 1 trials are completed. There are phase 2 trials ongoing. Just to summarize it, there was clinical activity, but as a single agent, there were only 2 CR, complete remissions, so where the patient actually, the disease was eradicated out of this 25 or 30 MLL rearranged cases that were assessed. There were many scenario responses where you would start to see the blast decrease initially, and then they would kind of turn around and start going back up. And we learned quite a bit in that trial. First of all, the responses all the responses took time. It often took many weeks to see the leukemia blast start to respond. We only got about 60 to 70% repression or suppression of gene expression. Interesting to think about what that means. And we developed a better understanding, both in the context of this trial and otherwise, that you need deep and consistent inhibition of this enzyme in order to get the phenotypic effects. And as usual, the thought is we probably need to find the appropriate thing to combine this with. And we continue to study the mechanisms of escape to DOT1 inhibition, but this is just showing you what cells will do. These are control cells growing exponentially here. You treat with the DOT1 inhibitor, they stop growing, some die, they sit there for a while, then they start taking off again. So they will adapt to the loss of a K79 methylation because it's still being inhibited DOT1 out here. So kind of what that adaptation is, we don't know at the moment, but continuing to study that. But this is a little bit of a predictor, I think, of what ultimately we saw in the phase 1. So we continue to work on DOT1, but about the same time, concepts were developing, as I mentioned, that inhibition of the interaction of MLL and menin might also be a relevant therapeutic opportunity because, show you here, the N-terminus of MLL, so these are the N-terminal amino acids of the MLL1 protein, which is in the fusion. These 5 amino acids bind to a pocket shown here in menin, and that interaction is critical to maintain leukemia development, first shown by Mike Cleary's group at Stanford. And so with that in mind, a number of chemists, first probe compounds developed by Yolanta and Beck at Michigan, but we then started working with a company at the time called Vitae to try to develop orally bioavailable, potent, and specific inhibitors that would bind to this pocket. And what's shown here is a protein or a small molecule we recently published called BTP-50469, bound into this MLL pocket. And the idea is probably obvious that if you bind the molecule to menin, you will kick off the MLL fusion, and that should lead to relevant changes in gene expression and maybe efficient proliferation. So this is just a summary of the IC50s for proliferation of this particular inhibitor on a number of MLL rearranged lines. Each of these down here, these 5 here are MLL rearranged as compared to HL60s, which are not. And you can see that these are really potent inhibitors of leukemia cell proliferation. And most other human leukemia lines that we've tested are not sensitive to the DOT1 inhibitor. I say most other because there are some with mutations in the NPM1 gene that I'll talk about in a minute that are sensitive to DOT1 inhibitor. So this is the molecule. I've already shown you this. So I told you that we thought what was likely would be happening when you treat with the menin inhibitors, you would disrupt the MLL-menin interaction. And we thought that might lead to menin being lost from the MLL complex. So this is just one of the experiments taking the first assessment of that. This is glycerol gradient centrifugation, showing menin here spread out over many fractions of the glycerol gradient. These higher numbered fractions are larger molecular weight complexes, and you treat with the menin inhibitor, and you see you collapse the menin down to what is very likely to be mostly free menin. So we kick menin out of whatever complexes, presumably the MLL complex, that it's associated with here. And that leads to gene expression changes that we can recognize as relevant. MEIS1 is a well-defined MLL target, PBX3 the same, MEP2C, a transcription factor we defined some time ago as a target and relevant in this disease. So leading to gene expression changes that we might expect. If you look at chromatin, so now what's happening at the chromatin level when you treat with the menin inhibitor? This is menin ChIP-seq. You can see that menin is lost from chromatin pretty much throughout the genome, so it's really stripped off of chromatin. Whereas MLL itself, at least at the global level, doesn't appear to change dramatically. That was interesting and maybe somewhat surprising. Most of the models would suggest that menin and MLL interaction should be critical for at least much of the MLL-associated chromatin binding, but this would suggest it's not. We actually found that you destabilize the DOT1 protein when you treat with the menin inhibitor. We don't understand yet the mechanism of that. And globally, H3K79 methylation didn't change. But if you look at specific loci, like the MEIS1 locus shown here, menin comes off of chromatin, MLL itself comes off of chromatin at this specific locus, DOT1 is gone, and this particular gene is one of the ones I just mentioned where gene expression is changing. Whereas the Hox locus, for example, menin's gone with the treatment, MLL is not, it's still there. There's still some DOT1, it's decreased. H3K79 doesn't change and gene expression doesn't change. So the point here is the MLL menin inhibitor is affecting a very selective gene expression program where a subset of genes where the MLL fusion is found. And where MLL is lost, you see a dramatic loss of gene expression. So this is a GSEA looking at RNA-seq and looking at the genes where MLL is lost from chromatin. So what we think this is telling us is that MLL disrupts the MLL-menin interaction. You get very specific and selective changes in gene expression. That leads to, I didn't show you this, but differentiation of the leukemia cells, and maybe the reason that we can treat mice with this inhibitor for long periods of time with minimal toxicity. So with this molecule that has very good pharmacokinetic properties, we're able to put it in mouse feed, makes experiments much easier. We can generate in leukemia in mice, either leukemia is derived, taken from humans with the disease, or our mouse models inject those into mice and then change the food, and you see the following. So the black are the control mice, and this is a mouse MLAF9, retroviral MLAF9 model, very aggressive. Anybody who's worked with this knows these mice will die relatively quickly. Here we've treated during this period of time with the BTP 50469 laden food, and it stops here, and in fact, the disease was eradicated from these mice. So this is an unusual degree of efficacy in this particular mouse model, and prompted significant number of studies in PDXs where human, in this case AMLs, were injected into mice, and where we saw also dramatic responses in the PDXs. We, at the same time, were teaming up with Richard Locke in Australia and Malcolm Smith at the NCI, who had developed a number of MLL rearranged ALL PDXs, as shown here. These are the translocations in the different ALLs. And this is a swimmer's plot showing you how long the mice survive after just 28 days of treatment here. So these mice, pretty long life for a mouse, 504 days, and a number of mice from this leukemia survived. There were variable responses in some of the others, but really pretty dramatic activity in ALLs. So to summarize that, this small molecule, and I'd say Yolanta Rebecca's group's been developing similar small molecules and similar types of activity, have really dramatic activity in mouse models and lead to very selective changes in gene expression and not much in the way of toxicity, at least in the mouse models. So while we were doing this, both DOT1 and menin, we were thinking about what other leukemias might be dependent on MLL or DOT1 or menin to see if similar mechanisms were playing out there. And we decided to look at another subset of leukemia that has high-level Hox gene expression. So this is gene expression data taken from a paper from Tim Lay's group showing the Hox gene expression profiles of a number of different leukemias, and this is the genetic characterization down here. So in green are the MLL rearranged leukemias. You can see HOX-A and MEIS1 expressions. That's pretty the definition almost of MLL rearranged leukemias. But if you look at this other big group over here, these are AMLs that either have DNMT3A, TET2, or NPM1 mutations. And this subset here, about half of them, have NPM1 and often DNMT3A mutations as well. So we decided to study the NPM1 mutant subset, and I'll tell you why in a minute, but the NPM1 mutation is found in about 30% of adult AML. So this is much more common, making clinical trials potentially more easy. And the mutant NPM1, mechanistically, what happens when the mutation occurs is the NPM1 protein now is in the cytoplasm where it's normally nucleolar. And mechanistically, how that leads to activation of gene expression, we're starting to look at. We don't know all the details of that yet. And these mutations usually occur or frequently occur with other mutations like DNA methyltransferase 3A. So Michael Kuhn, when he was a fellow in the lab when we lived in New York, had done a CRISPR domain screen that was initially an approach initially developed by Chris Vakich's group in NPM1 mutant cell lines, and found that the domain of MLL here that binds to menin, when you mutate that with CRISPR, leads to a pretty impressive loss of proliferation of the NPM1 mutant cells. And went on to show that with the kind of first generation menin inhibitors, that indeed it looked like NPM1 mutant AML was relying similarly to MLL on the MLL menin interaction. But this is presumably wild type MLL that it's depending on, not diffusion. At about the same time, the concepts John Dick's group and Irv Weissman and Ravi Majetti were looking at the various populations of hematopoietic cells in patients with leukemia, and it's shown that in the situation where you have DNMT3A and NPM1 mutations, the DNMT3A mutations are very likely, and this is borne out over many studies now, to be in the hematopoietic stem cell, and that's probably the first mutation in progression to AML. But the NPM1 mutations look like they're most often in various progenitor populations and maybe not in the stem cells. And that led us, Hannah Uckelman, a postdoc in the lab, to start to address the possibility that the DNMT3A mutations, which are most frequently found in this syndrome called clonal hematopoiesis that many of you may know about, where as we age, we'll develop stem cells that have mutations frequently found in AML, but that in of itself does not necessarily lead to the development of leukemia. But if you acquire other mutations, in particular NPM1 mutations, then you're almost guaranteed at that point to go on and develop leukemia. And the question was, is this perhaps happening in the progenitor population and activating that stem cell program much like the MLL fusion? I won't go through all the mouse studies, but just to summarize here, she obtained genetically engineered conditional mouse models where we could activate DNMT3A from George Vasilio's group in Cambridge, or NPM1 from Ross Levine's group at Sloan Kettering, crossed them, made all the appropriate crosses, and was able to show that as you move down hematopoietic development from a stem cell to a more committed progenitor, this is called a multipotent progenitor, to a common myeloid progenitor, to a granulocyte macrophage progenitor, the Hox proteins genes are being shut off. We knew that from previous studies. But when you have an NPM1 mutation, plus or minus the DNMT3A mutation, that program is unable to be repressed to the extent that it is normally. This is similar to what's happening with the MLL rearranged leukemias. And if you look more broadly, it's many of the Hox genes that are behaving this way, and in fact, many of the genes that are in traditional stem cell signatures. So the NPM1 mutation appears to be maintaining this stem cell gene expression program in the progenitors. And just to be thorough, she did the experiment where she activated the NPM1 mutation in progenitors, not actually, so isolated the progenitors, activated NPM1. That led to immortalization of cells and serial replating. That's how we assess that. And ultimately, those cells would self-renew in recipient mice for long periods of time. So essentially what she generated was a GMP that was self-renewing to some extent, like a normal or like a long-term hematopoietic stem cell. these these mice would ultimately get leukemia, but there was a long period of time of this extensive progenitor self-renewal before the mice ultimately developed AML. So this tells us that progenitors can act as the cell of origin of NPM1 mutant AML, just very similar to what we saw with the MLL fusion. So we think what's happening in this disease is DNMT3A mutations come first. That's very likely, most often the case, in a hematopoietic stem cell. That gives rise to progenitors, all of which have these DNMT3A mutations. And then in some downstream progenitor, the NPM1 mutation arises. That leads to activation of a stem cell program in the wrong cell type, and including the HOX-A, and in this case also the HOX-B cluster genes, and MEIS1, which is critical. I'll show you in a minute. And that really then, prompts us to think about NPM1 mutant AML as another candidate for treatment with whatever small molecules we're developing for MLL rearranged leukemias. I won't take you through this again. It's probably obvious. We now went to our MLL menin inhibitor to assess what activity it might have in this leukemia. So this is a human leukemia cell line. OCI-AML3 is an NPM1 mutant cell line. And you can see here that DTP50469 has a very potent anti-proliferative effect on this cell line. These are the mouse leukemia cells. You see we can inhibit serial replating activity or colony formation with the small molecule. And primarily the phenotype is differentiation. If you look at gene expression, it looks very much like what we saw with MLL. You lose with the MLL rearranged leukemias, you lose MEIS1 expression, you lose PBX3 expression. The HOX genes change a bit, but not dramatically. And that's really something we see both in the MLL rearranged setting and NPM1, is that the HOX genes will change a bit now and then, but they really never change to the same extent or as rapidly as MEIS1, PBX3, and some of the other targets do. This is just an experiment to show that MEIS1 is important in this disease. That was not so clear when we were initiating these studies, but if you CRISPR out MEIS1 in the mouse NPM1 mutant cells, they'll stop proliferating. And importantly, we can actually rescue the sensitivity to the inhibitor by expressing MEIS1 off of a retroviral promoter, so taking the MEIS1 out from under the control of MLL and menin. So this change in MEIS1 expression, we think, is quite relevant for this disease. To summarize this quickly, in the NPM1 mutant setting, if you look at chromatin, it looks very similar to what's going on with the MLL rearranged cells. That is, MLL itself is lost at selective loci like MEIS1. Those are the loci, or those are the genes expression decreases, and it's not at others like the HOX-A cluster. So this is a common theme between the MLL rearranged AMLs and NPM1 mutant AMLs. So that led to two different types of in vivo experiments. The first one is shown here, which is a mouse experiment that I alluded to a few minutes ago, where you can take granulocyte macrophage progenitors, activate NPM1, inject those into mice. They'll self-renew for long periods of time, but you won't get leukemia. You inject them into secondary recipients, and then ultimately over time, the mice will develop leukemia. So we wondered if perhaps if we treated with the menin inhibitor prior to the development of full-blown leukemia, could we prevent the progression to AML from that self-renewing progenitor population? And this is just showing the engraftment of these cells in mice, so the presence of the cells in mice over time in the control-treated mice versus the mice treated with the menin inhibitor. And in fact, you can see we pretty easily eradicate these pre-leukemic or cells in the mouse model, and that did indeed prevent the mice from developing AML. So at least in this model, the progression from self-renewing progenitor to full-blown leukemia can be prevented by treatment with the menin inhibitor, and again, as I said, with very little toxicity. This is showing two PDX model experiments. These are human leukemias now growing in immunodeficient mice. This one's an NPM1 FLT3 ITD double mutant AML. This is a very aggressive disease in people. And you can see this during the gray bar here is the period of treatment. And those mice were all cured of their human leukemia. And we even did this in a much more, raised the bar for this molecule and started treating very late in the the development or progression of the disease in the mouse model. And we were even starting treatment a few days before the control mice died, we were able to rescue many of the mice. So this is really a pretty impressive response. We'll skip that and now talk a little bit about briefly about where these menin inhibitors stand in clinical development. So the phase 1 trials, both a version of the molecule that I've been showing you data from that developed by Syndax and a molecule developed by Cura Oncology are in phase 1 trials in relapsed refractory leukemia based on preclinical data from our lab and from Yolanta Rebecca's group. And at last AACR, Syndax presented some of its initial patients that were treated, in this case with relapsed refractory MLL rearranged AML. And the ones at that point where they had gotten to the plasma concentration of the small molecule, there was only one patient at this time, the plasma concentration of the small molecule they thought would be required for a response, that patient went into a complete remission and with the only toxicity being a grade 2, so a modest effect on the EKG or QTC in this patient, which resolved by adjusting the dose. So that was really the first evidence that MLL rearranged, this is adult MLL rearranged AMLs could respond, and they're going to report an update on this, I think, in the next few months. Many more patients have been treated now. At ASH, Cura Oncology presented some of their initial data from their phase 1. I won't go through all the details. It's a standard phase 1, similar to Syndax phase 1. But in this case, they had a few different types of AML and 2 NPM1 mutant AMLs that had relapsed. And one that's of particular interest is this patient with the DNMT3A mutation and NPM1 who had been through 7 different combinations of treatments and was still refractory. And this patient went into a complete remission with no detectable minimal residual disease. This is a pretty impressive remission. And here's another patient that had a pretty impressive response as well, who had a FLT3 ITD. Point being early, but looks like these molecules indeed may have activity and not, again, significant toxicity. So I'm going to stop on this part here and summarize now in quickly in the next 10 minutes or so, kind of moving out of leukemia. But so the question often arises is, well, why is it, do you think, that the menin inhibitor seems to be, at least it would suggest, we'll see, more effective than the DOT1 inhibitor? And we've started looking at that a bit, and many caveats to this statement, but one thing that appears to be the case is when you inhibit the enzymatic activity of DOT1, you can get this complex off of chromatin at specific places, but it takes a long time and it's not so consistent or dramatic. Whereas when you inhibit the MLL-menin interaction, the MLL fusion comes off rapidly, you destabilize many of these other proteins. So it's a much more dramatic, if you will, disruption of the MLL complex than DOT1 enzymatic inhibition. So we think, and the changes in chromatin and gene expression are much more rapid. So that is still hand waving, but we think that has something to do with the difference. So I'm going to end the data part here with just kind of shifting gears and addressing the question as to whether or not any of these concepts play out in other diseases. And certainly those of you at St. Jude have heard from Charlie Roberts and others about this disease and the complexes associated with it. So synovial sarcoma is another cancer, sarcoma, that's found in adolescents and young adults. It has a recurrent translocation here, X18, that it leads to the production of a fusion protein where SS18 protein is fused to one of these SX proteins. The relevance of that in this context is that SS18 is normally found in the so-called BAF or switch-SNF chromatin remodeling complex, which is a large chromatin-associated complex that remodels chromatin to control gene expression. We, a number of years ago, again, back when we were working with Chris Vakich's group a fair amount, did a CRISPR screen, again, a domain-focused CRISPR screen, in a number of sarcoma lines and found that guides that targeted the bromodomain of a protein called BRD9, which resides in that complex, were particularly, or inhibited pretty dramatically the proliferation of these synovial sarcoma cells, 2 different cell lines. Whereas those guides had minimal effect, almost no effect on the proliferation of other sarcoma lines. And as more CRISPR screens have been done, this has held up that BRD9 is a selective dependency in synovial sarcoma, rhabdoid tumors, as recently shown by Charlie's group, and has some of a dependency in AML as well, but most other cell lines don't seem to care. And Jerry Bryan, the fellow who was doing this, wanted to make sure that BRD9 was actually in the fusion protein associated complex and did IP mass spec here with antibodies to the fusion protein. And suffice to say, BRD9 was indeed in the complex. If you look at the stoichiometry, we were already guessing that it probably wasn't in every complex. And now, again, work from Sigal Kadosh and Charlie and a number of people have shown that indeed this BRD9 containing complex is somewhat different than the more traditional complex. But it is there and it is a dependency. And fortuitously, we were in the process of developing working with Jay Bradner's group at the time to identify small molecules that lead to direct targeted protein degradation of BRD9. So many of you may know about this recent chemical approach to target the ubiquitin machinery to proteins of interest as a way to selectively degrade them rather than inhibit them. There are a number of ways to do this. One that's used quite often is called a PROTAC approach, where you have a small molecule that binds to the target. In this case, it's going to be a BRD9 bromodomain inhibitor, and a small molecule that recruits an E3 ligase, part of the ubiquitin machinery. In this case, it will be a thalidomide-like molecule, and you fuse them together to essentially target this ligase to that protein, and that leads to degradation. And to make a long story short, we developed we in this case is Jay's group, developed 2 small molecules that lead to really impressive degradation of BRD9 in all cell types. These are synovial cells. And that really also leads to impressive inhibition and proliferation of both of these synovial sarcoma cell lines. And interestingly, in black, this here is what you see happens to proliferation if you just treat with the inhibitor. So the inhibitor itself, which binds to the bromodomain of BRD9, doesn't have much activity, but when you make that a degrader and degrade BRD9, now you have pretty dramatic anti-proliferative activity. And these degraders have no effect on the proliferation of other sarcoma lines. And I tell this both because I think mechanistically it means there are similar things, different complex, but similar concepts going on in this disease as in MLL-rearranged leukemia, but also these types of molecules are probably going to enter the clinic sometime in the next year from a couple of different companies that are developing them. So it'll be interesting to see in this case as well how this approach plays out in patients. And just to go back to the concept that I presented between DOT1 and menin, we think something similar may be going on here. That is, you have an inhibitor of BRD9, this large multi-protein complex, and there's probably multiple ways that the complex is associated with chromatin. And that in of itself doesn't appear to be enough to do much in terms of proliferation, in terms of chromatin association. But if you degrade BRD9, now the complex, we think, is likely to be much more unstable and certainly much less likely to bind to chromatin. Much like we saw with the menin inhibitor, this is a much more dramatic way to perturb the complex. We're not digging into the mechanisms of this in any particular detail. We'll let Charlie and Sigal and others do that. But I think from a mechanistic and conceptual standpoint, we are starting to see that you probably need to be somewhat aggressive when targeting these complexes and not just inhibit one activity in a complex of 10 or 15 proteins, you probably need to destabilize the complex much more dramatically than that. So to summarize what I've told you here, we started with MLL fusions. We still work on that significantly. That led us to a number of potential proteins to target a number of different small molecules. We now know that NPM1 mutant AML is also sensitive to these approaches. That actually accounts for about 35% of all adult or all human AMLs, and we're starting to look at other subtypes as well. We think some of the concepts we found in AML are playing out in synovial sarcoma as well, and recent studies have shown that the Ewing sarcoma fusion may be using some of these complexes as well. And I mentioned the VAF complex in this sarcoma story, but certainly in this setting or in this group, I would be remiss to not point out that there's a lot of work being done on the PRC2 complex as well, and small molecule inhibitors of the EZH2 enzyme have shown significant activity in rhabdoid tumors, epithelioid sarcoma, and lymphomas, based significantly on Charlie Roberts' work showing that this EZH2 becomes a dependency when the VAF complex is genetically perturbed. And then this is the last slide I'll show, just because I find this so fascinating, that a number of groups, Susie Baker at St. Jude, Nada Javato at McGill a number of years ago, identified mutations in histones themselves and in the lysines, like lysine 27, that are modified with, in this case, methyl groups, or in amino acids near the lysines that are modified. Those are the drivers of very aggressive forms of pediatric brain tumors, and the type of mutation highly correlates with where in the brain the disease will develop. So this is really clear evidence that, one, epigenetic processes are central, as I've been talking to you about for the last hour, and that, two, this is clearly development gone awry, which just makes so much sense in the setting of pediatric cancer. So I hope I've convinced you that we are starting to understand chromatin and epigenetics enough to think about targeting it with small molecules. The toxicities to date have not been particularly dramatic, something that we were all worried about as we started in this field. We'll certainly need to keep an eye on that as we move these molecules into younger and younger patients. But at the moment, anyway, the toxicities have not been tractable. So I'll end there and think this is a pre-pandemic picture of the lab. Hopefully we'll be able to get together and take pictures like that again soon. A lot of tremendous fellows and students and technicians have driven this work. Here are some of our collaborators, funding agencies, and I'll also point out that we really appreciate the funding from St. Jude through one of the collaborative programs that they've developed focused on chromatin. So I will end there, and I got us in just under the wire.
Thank you very much, Scott. That was fascinating. It's really it's amazing that these multi-component regulatory complexes have these vulnerabilities with respect to particular components, either inhibition of interactions or degradation. Thank you for that. There are quite a number of questions, so we'll field questions for a few minutes, and then we can enter into an extended overtime period. So the first question is from Bingqing Zhao, and it is, in the .L1 inhibitor trial, while the drug takes time to take effect, are patients under any kind of other treatment?
Yeah, most of it. Well, in the setting of a phase 1, you have to limit the other types of treatments that patients can receive. But in that particular trial, they were able to receive hydroxyurea, which is a treatment that will keep the leukemia at bay to some extent. So that's why we were able to treat patients for a longer period of time and see what their responses might be.
Okay, thank you. Next question is from Konstantin Milonas. First of all, they say great talk. Menin also interacts with MLL3 and 4 over poised enhancers. Have you looked at MLL3-4 distribution and H3K4 monomethyl levels following VTP treatment?
Yeah, so menin most famously also interacts with MLL2, which is another promoter-associated complex, and we've looked at that a bit. We're just starting to look at MLL3 and 4, but don't yet know what menin does in that context. And, kind of alluding to the fact that menin probably is in other protein complexes as well, which has been shown by a number of groups, and indeed we might be having effects beyond just MLL1 and 2, but more to be done.
Okay. Next question is from Bapaditya Chandra, actually a postdoc in my lab. Thanks, Bapa. So for the MLL fusions and as well as the NPM-C mutants, when the compound VTP is incorporated, it inhibits interactions with menin and at the relevant chromatin sites. So he's asking about really how release of menin is then responsible for, inhibiting changes in gene expression and the effects on leukemic cells.
Yeah, so it's needless to say that's an important question. So what we see, and I'm not going to be able to give you all the molecular details, but is that at those loci where gene expression changes, you lose the MLL and a number of other, most of the activating, if you will, chromatin complexes. So the chromatin assembly there really falls apart. So that must mean that MLL and menin are playing an important role in maintaining the chromatin in a state such that those complexes can remain associated. And we don't lose them at the places where MLL doesn't come off. And so the belief there is that MLL must be using a different adapter protein, which is what we think menin is, to remain associated with chromatin in that particular at those loci. Obviously, to prove that, we would need to show what that adapter is, and we haven't found it yet. But that's the current hypothesis.
Great, thank you. Papa had a second question, but out of deference to others, I'll move on. But before I do that, so it's now 1:00, and so formally the session has ended, but Scott has graciously agreed to stay on for additional questions and discussion. Before I move to that, I want to first announce the speaker for next week. Next week's Science of Childhood Cancer lecture, and the speaker will be Robin Yabroff from the American Cancer Society, and he will discuss financial hardship after childhood cancer. So that will be a week from today, February 18th at noon Central Time, 1 p.m. East Coast Time. And okay, so now on to further questions. So this question is from Song Kao. First of all, they say very exciting results. So a general question for fusion drivers like MLL fusions, is a high expression of MLL, I guess they mean the wild type protein, required for tumor development, or the underlying different structure from wild type due to the fusion is important for tumor development? So I guess the question is there a role for wild type MLL in sort of the takeover by the fusion protein, or is the fusion protein's aberrant function really distinct and solely responsible for the phenotype?
Yeah, it's a good question, and the answer to that has been a little bit muddy over the past 10 or 15 years. It has certainly been looked at in quite a few different directions. And what it looks like, at least in the mouse models, is that, so if you make a mouse model where you can genetically inactivate the the MLL alleles, both of them, since you're putting in a retrovirus, the wild-type MLL alleles are not required for MLL fusion to continue to produce leukemia. So at least in the maintenance situation, once the leukemia is developed or the cells are transformed, the wild-type MLL is no longer necessary. It doesn't mean wild-type MLL might not be necessary for the initiation of the disease, but that experiment's just harder to do because it's harder to set it up. But for maintenance purposes, it doesn't look like it's required.
Okay, thank you. Next question. So there's maybe a half a dozen more questions, at least so far. So next one is from Kyla Tooley. Given the DNMT3A mutation, have you looked at the DNA methylation profile of these cells before and after treatment with the menin inhibitor?
Yeah, that's a good question. And the answer is no, because we don't know yet whether or not the menin inhibitor or MLL1 is playing any role in the DNMT3A phenotype. We know it's playing a role in the NPM1 phenotype, but we don't know about DNMT3A. So that is a good experiment for sure. I think we just need to get a little bit more evidence that DNMT3A is working via some of these mechanisms first.
All right, moving on. This is from Monica Balmer. So when treating with BRD9 PROTAC, did you ever see degradation of anything that's in complex with BRD9? Interesting question.
Yeah, it is a good question. And I'll have to say, unfortunately, we haven't looked too closely, but I know what. So there are two reasons you might get degradation of other complex members, one of which is you just break apart the complex and the proteins become unstable as a result of the complex falling apart. The other is the question as to whether or not you get any bystander ubiquitination as a result of PROTAC. And at least as far as I've heard, and I've asked this question of many people developing PROTACs, that I don't think that bystander type ubiquitination is very common. So as far as so that's a long, I didn't directly answer your question, but I think I got at what you were implying. So I think it's unlikely that we would get that type of direct degradation as a result.
All right. Irina Viahova says, thank you. It was a great lecture, but didn't ask a question.
I like
so Judith. There's actually a couple of those. So good job, Scott. So Judith Baer does have a question. So the question is, the menin inhibitor shows great activity in the preclinical and early clinical data with prolonged responses both in mice and patients. What would be the reason that leukemic cells apparently cannot get resistant to this type of drug?
Well, so we can make leukemia cells that are resistant to this drug, so that it's not impossible. But it is true, even in the tissue culture, it's harder to make resistance to this molecule than it is to DOT1 inhibitors, for example. And I'll hand wave as to why I think that's the case, but we haven't really directly shown this. I alluded to this in the talk that the changes, the chromatin-associated and gene expression changes are much more rapid when you treat with the menin inhibitor. Like 48 hours, everything's falling off, chromatin gene expression changes are happening. The DOT1 inhibitor can take 7, 10, 12, 14 days. So I think not a very molecular answer is that the pace at which you are disrupting these complexes probably matters in terms of the way that the cell can adapt. And so it's just harder for that patient to happen that rapidly. And we're looking at these things, but it's difficult to prove that. And the other thing I'll say is that the menin inhibitor also leads to loss of the superelongation complex at these genes. So it's really disrupting chromatin pretty dramatically.
Okay, next question from Chao Yi Yang. Given the example of menin inhibitors, do you think targeting other adapter proteins in the MLL complex to destabilize the complex is a viable approach to target that MLL complex. And an example is the menin inhibitors do not clear the MLL complex binding to Hox genes.
Yes. Yes, I think the answer is that is likely to be the case. I mean, another way to ask that question is, what is it that's keeping MLL at the Hox genes whenever menin is gone? So whatever that is would likely be relevant. I mean, there are molecules that target WDR5, which is another component of at least the wild type MLL complex. Those have pretty broad activity in terms of gene expression. So I think the potential problem is with any of these is in some cases the proteins are going to be selectively required for certain gene expression, and in some cases they're going to be required broadly for gene expression, and that's probably more difficult to deal with from a therapeutic standpoint.
Okay, thank you. Next, from Milad Rasuli. Thanks for a nice talk. Have you ever checked the menin inhibitor on other AML types that express high levels of Hox genes, like NUP98 fusions? So, well, I guess you checked it with the C+ mutants of NPM1, but what about the NUP98 fusions?
Yeah, we're as I think you know, Richard, we're looking at that. And early data suggests, and it is still early, so I don't want to be too definitive about this, but it does look like those cells are probably sensitive to the menin inhibitor as well, and you see changes, what look like relevant changes in gene expression. So maybe not terribly surprising, but it looks like it could be the case there as well.
Okay, good. So we seem to be at a steady state of about a half a dozen questions. So we'll go. You're on the hook for a little bit longer, Scott, if that's okay with you.
It's okay.
So Naftai, oh God, I'm going to butcher this. Nira Manzi, sorry. Thank you for the presentation. How do you consider the safety and therapeutic efficacy of these chromatin complexes in treating cancer? Do you plan to use them for other different types of cancer?
So, I mean, I think for now we'll try to develop them in the places where they make the most sense, and where it looks like there is a significant therapeutic index. I thought. And other cancers, I mean, the menin inhibitors themselves have been looked at in a number of different cell lines, as you might imagine. And outside of hematopoietic malignancies, it's not been so obvious that there is activity there. There's still people digging into this for sure, but so it's not obvious to me anything outside of heme malignancy would be immediately a place to develop them.
Okay, next is from Jules Mayering. Who says, thanks, Scott, great talk. And they ask that, would you think that other HOXA driving events, so this is sort of a question along the lines of a past one, you think that other fusions that drive HOXA family gene expression, such as SET, NUP214, PI-CALM, MLL T10, or DDX3X, MLL T10, that also recruit DOT1L, may further depend on menin and therefore be sensitive to the VTP inhibitor?
Yeah, I think similar to what we saw with NPM1, I think those are all reasonable. I mean, we obviously haven't tested them all. The models are not great for all those various types, but to date, anything where we've had a model that we could assess that had a high level Hox gene expression, there seemed to be a subset at least of those leukemias that were indeed sensitive. And the other thing I'll point out that anywhere you have sensitivity to a menin inhibitor, at least as far as we've studied, and if you look on the Broad dependency map, it's the same for genetic inactivation, you have sensitivity to DOT1. So those almost overlap completely in their dependency profiles.
Okay, we're approaching the end. Lawrence Vandermeer asks, the existence of a leukemic cell in ALL is a leukemic stem cell in ALL is subject to debate. Many cells seem to be leukemia propagating. How does this relate to the seemingly reduced sensitivity to menin inhibitors?
Well, I think what's being alluded, so I agree with the first statement there that I'm not sure there's certainly heterogeneity in ALLs. Whether or not there's a leukemia stem cell the same way there is in AML, that's still a question. I think what may be being alluded to is that the ALLs in the DOT1 trial, the patients that had MLL rearranged ALL didn't have the same types of responses as patients that had AML, leading to the question as to whether ALL was different. Where we've tested ALLs in the PDXs with the menin inhibitor, they've shown similar types of sensitivity to AML. So I don't know there's any reason to necessarily predict that ALL is going to be less sensitive to menin inhibitors, but we're going to find that out hopefully in the next year or so.
Okay. Thank you. So Fupan Yao asks, basically they ask whether you've applied any single cell technologies in your studies. So you've used, sort of bulk cell methods to probe, the binding of various complex components to chromatin and so on. So I think they're asking, have you done any single cell studies, and would they provide any sort of unique insights into the function of these complexes in leukemia?
Yeah, that's the perfect way to ask that question, meaning, have we done it? And if we did do it, would it be something that you should have done? So you can probably see my bias on that. I mean, yes, yeah. I'm sure. So we have started doing those types of experiments just because you kind of have to. But I'm not entirely sure what we'll get from it. But maybe that's the best experiment to do. Just do it and see what happens. But we did single cell RNA sequencing on one of or a few of the ALL PDXs that responded, and most of the cells responded the same way that we saw in the bulk. So there wasn't any obvious heterogeneity in response, but we haven't looked at it in great detail yet.
Okay, we're getting toward the end. So from Chao Yi Yang, do the menin inhibitors change the clonal evolution besides just killing leukemia cells?
Yeah, that's a great question. I don't know the answer to that, but we're certainly, we can look at that both in the clinical trial and in the PDX models. And that, I'm making jokes about single cell experiments, that particular question is better done in single cell type experiments. And indeed, we're going to do that.
Yeah, most certainly. Great. So Giles Robinson at St. Jude asks, he asks, in addition to fusions, there are many frameshift or nonsense mutations in the MLL genes in cancer. Do you have any idea as to what these mutations may be doing in these cancers?
Yeah, that's a good question. Those what are presumably loss of function mutations are most commonly in MLL3 and 4, which are the MLL family members associated with enhancers. So I suspect, even though the data is not particularly good yet, that those are, of course, probably perturbing gene expression via regulating enhancers, but probably a different mechanism than we're looking at here. And obviously, as a result of them being loss of function are behaving as tumor suppressors rather than oncogenes, which the fusions appear to be doing. So it's a long way of saying, I don't really know, but it's interesting, but it's probably different than what we're looking at.
Okay, thanks. Okay, the next to last question. So great talk again. Have you looked at how the menin inhibitor affects the structural or organization of nucleosomes. And what are your thoughts on that?
The structural organizations of nucleosomes.
So I guess, nucleosome occupancy on the sites engaged by these complexes.
Yeah, we have not looked specifically at that. Short answer is I don't know, but I think I alluded to, we have looked at it with the DOT1 inhibitor. The same's probably happening at the MEIS1 locus, for example. When we treat with the DOT1 inhibitor, that whole locus is just, there is nothing left there that is an active modification. So I think that we see K9 methylation starting to coat. So I think you're getting something forming that is the beginning of some type of heterochromatin formation, but we have not looked at that in great detail. So I suspect that everything's getting compacted, but we don't know for sure.
Okay, and now to the final question. So this is from Waitman Allman. Again, he says, assuming it's a he, great talk. You've discussed the specifics of menin inhibitors, DOT-L1 inhibitors, and there are many other types of small molecule inhibitors. What are your thoughts on where we are going in this field more broadly? Said another way, do you foresee a future where we are able to have such focused therapies moving away from traditional chemotherapies? It's a very good question. Are these targeted therapies going to be successful such that they really are the future of cancer therapy?
Yeah, that's a terrific final question.
It is really. Thank you for that.
So I would say we have an example, acute promyelocytic leukemia, ATRA and arsenic combination that target the fusion protein in that case, destabilize the fusion protein in that case, have essentially supplanted chemotherapy as a treatment in a disease that used to require a bone marrow transplant. So even beyond imatinib and the kinase inhibitors, there are examples where targeted therapies that target the right, really get at the heart of the action are able to replace chemo. Will these molecules that I talk about be those? Who knows? Ultimately, obviously, I think what we want to do, and I think we're all trying to figure out how to do this, is directly target the fusion proteins and degrade those. But that's, at least for the MLL fusions at the moment, not possible. But so the answer is yes. I think we'll get there. Hopefully, we get there during my career.
Yeah, there are some years left, Scott. It's good to be positive. All right, so that's the end of the questions in the chat box, and so I will call it there. So that it then falls to me to thank Scott once again for a really stimulating lecture that really shows the future of targeted therapies against a variety of especially pediatric leukemias as well as adult leukemias. Thank you to everyone who attended. At the peak, Scott, there were slightly over 300 people, and there's almost 90 still on the call listening to you answer questions. So thank you very much for staying around. And I guess that's all we have for today. So thanks everyone again, Scott.
Thanks, Richard.
Attendees, and we'll see you another time.
All right. Thank you.
Now.
Bye.